Design for a volumetric microfluidic chip. An overview of the proposed device and a simulation using Jell-O. Materials: styrofoam bowls, coffee stir sticks, Jell-O, vinegar, baking soda, and food coloring. The proposed device. This slide is a close-up of the design. The lane that is running down and diagonally is the lane where the sample will be washing along the device and connects all testing lines. The upper lane that runs upward diagonally is reaction lane, where we will expect a chemical reaction to initiate within the well. The bottom upward sloping lane is the control lane. Pictured here is the top view of the chip. There is an inlet in the top plate in order to manually load the sample using a pipette in the bottom left corner. There are also outlets at the end of each lane. The outlet of each channel ensures that the driving pressure force is able to flow along one direction from an area of high pressure to an area of low pressure. This prevents the gas from finding an equilibrium state. What does the device do? It detects abnormal levels of HER2, a protein commonly found in some types of breast cancer. How does it work? Enzyme-linked immunosorbent assay, or ELISA, is a diagnostic tool that can allow a sample to be diagnosed for a particular biomolecule. In our device, HER2 molecules are bound by capture antibodies that have been added to the surface of the microchip. The capture antibodies retain the HER2 until a second antibody detects it. In this device, the secondary antibody has a catalase probe that initiates a chemical reaction that produces pressure to drive fluid motion. The pressure-driven reaction is a combination of the catalase found on the target antibody and hydrogen peroxide. This gas-forming reaction produces oxygen. The pressure associated with oxygen will then pump fluids through the chip. In the Jell-O simulation, the gas-producing reaction is simulated using baking soda and vinegar. Baking soda represents the catalase. The reaction in simulation is acetic acid plus bicarbonate, which produces carbonic acid. Due to the instability of carbonic acid, water and carbon dioxide are produced. As you can see, the sample and control are found near the channel. From the left, the first vertical channel is the test channel, and the second is the control. Colored vinegar is added. The reaction that occurs drives fluid into the test channel. When the fluid hit the base of the control channel, no reaction occurred.